Greetings, this is Ignacio Camarillo, and I'm a faculty in the Department of Biological Sciences at Purdue University. My talk is entitled Use of Quantitative Proteomics to Define Mechanisms of Lippia origanoides in Activating Apoptosis in Triple-Negative Breast Cancer Cells. So I want to say that I'm very thankful for this opportunity to share some of our work and to highlight some of our studies that have been focused on this plant extract and defining its anti-cancer properties. So my talk will be divided into a couple major components. First, I'll talk about the overall background in triple-negative breast cancer. I'll pinpoint some identifications of triple-negative breast cancer vulnerabilities for potential future development of therapies. I'll give a short perspective on some plant compounds that have been very important in the development of cancer treatments. And I'll focus in on Lippia origanoides, which is a plant that we've been studying for a couple of years that we've shown that has some, it's a potential source of anti-triple-negative breast cancer compounds. I'll focus on our current hypotheses and specific aims, and then share some of our results that we've uncovered regarding the impact of the Lippia origanoides on triple-negative breast cancer cell apoptosis and some mechanistic insights of Lippia. So breast cancer is a leading cause of cancer-related incidence and mortality worldwide. So some recent figures show that breast cancer is the second leading incidence of cancers worldwide. So it is responsible for 11.6% of all new cancer cases. And this is considering both sexes, all types of cancer and for all ages, worldwide. Breast cancer is also responsible for a high number of cancer deaths. In particular, 6.6% of all cancer deaths are attributed to breast cancer. What's of a significant concern, substantial concern, is that breast cancer incidence and mortality are anticipated to increase by 50% within the next 2 years. So here's a table of the several different types of breast cancers that can be identified at diagnosis. Okay. So you'll see here that luminal A, luminal B, HER2 overexpressing and normal-like expressing, for the most part, have different types of receptors that are expressed, within them, and they have the specific markers, and they're, generally more responsive to therapies than the more aggressive basal, triple-negative breast cancer cell, which is characterized by an absence of estrogen receptor, progesterone receptor, or HER2 receptor. When we look more closely at the distribution of cancers, at diagnosis, we see that the majority of the time, the cancers that are found in patients are of the normal-like, luminal A, luminal B, or HER2. So these are generally more treatable. However, this basal-like triple-negative breast cancer accounts for 12.3% of cancer diagnoses, and they're definitely more aggressive. They are associated with decreased overall survival compared to hormone receptor positive or HER2 positive breast cancers. They have an increased risk of death within the first 2 years of diagnosis, the highest 3-year relapse rate compared with increased metastases. And patients with metastases have a survival rate, or the duration of survival for metastases patients with triple-negative breast cancer is only half of a year. So, what are some of the risk factors that are associated with triple-negative breast cancer, TNBC? Well, in the United States, it's been shown that African American women of African American descent, have a twofold higher risk of getting breast cancer compared to other ethnic groups, white, Latino, or Asian Pacific Islander. Specific mutations in genes are also associated with increased TNBC risk. So in particular, an overall risk is much higher for getting TNBC when there's mutations found in several genes, including the BRCA genes. Other risk factors for TNBC include usage of oral contraceptives in women that are 18 years of age or younger. TNBC risk is higher in younger women. Women that are less than 40 years of age are disproportionately diagnosed with ER-negative breast cancers compared to older women. And women that are obese have a 2.3-fold overall higher risk of getting breast cancer than non-obese women. Some good news is that breastfeeding tends to reduce TNBC risk. So Paris women that breastfeed for at least a year have a 31% reduction in TNBC. And in particular, African American women ages 20 to 44 who breastfeed at least 6 months have an 82% lowered risk of TNBC. So the big challenge with the TNBC treatments is there's not, very effective targets or treatments, for addressing this aggressive cancer. So one of the big sort of efforts right now is to identify, specific targets for TNBC, and that starts with really highlighting what are some of the vulnerabilities of TNBC. So at this point, we know that there's a constitutive activation of NF-kappa beta signaling that promotes survival and progression in TNBC. Early studies by Nakshatra et al. Showed that NF-kappa beta, so this transcription factor, right, is present at high levels and is very active in cell lines, breast cancer cell lines that are hormone receptor negative. Right? So these two cell lines are breast cancer cell lines that are estrogen receptor positive, and the rest of these are estrogen receptor negative, and you can clearly see that there's an upregulation of this transcription factor, okay, compared to ER positive cell lines. So how does this NF-kappa beta signaling system work? Well, the NF-kappa beta signaling is really regulated through an interaction of TNF-alpha with its specific receptor. So its receptor, once it's activated, can associate and activate two different complexes. Complex 1 leads to activation of the NF-kappa beta. In this case, NF-kappa beta is designated by this complex P50RLA. And in this case, right, the more aggressive cancers have all these components, right, that are increased in levels of protein and their activity. So what happens is once TNF-alpha receptor is activated, this interacts with the receptor interacting protein and then activates a series of kinases, which then ends up phosphorylating an inhibitor of NF-kappa B. So this IKB is an inhibitor of kappa B. When this gets phosphorylated, right, it gets signaled for ubiquitination, which then is degraded. Once this inhibitor is degraded, NF-kappa beta is free to enter the nucleus and then regulate gene expression, particularly genes that are associated with cell survival and proliferation. So the CIAP2 and Cyclin D1 and CFLIP. So in normal cells, right, this signaling pathway is not overexpressed, and TNF-alpha regulates this complex 2, which leads to activation of caspase and then caspase 8 and then apoptosis. Okay. However, in more aggressive cancer cells, when this complex 1 is activated, not only does this promote proliferation and survival, but it also activates genes that suppress apoptosis. So this is a dual effect of this NF-kappa beta system in the aggressive cancer cells. So we know that TNBCs often display aberrant expression of NF-kappa beta proteins, and that targeting NF-kappa B in TNBCs has been shown to reduce proliferation, invasiveness, and induce apoptosis. So there's other substantial changes that occur in aggressive cells such as TNBC cells that characterize and lead to its difficulty in treating these cells or this type of cancer. And it's known that there's different waves of metabolic reprogramming that occur in aggressive cancers that lead to their resistance to therapies. So it's known that TNBCs can increase their use of different alternative energy sources and other pathways to promote their survival. So they increase their fatty acid oxidation and membrane lipid synthesis, which is important for energy production and production of membranes for the rapidly dividing cells. These cells also initially go through this Warburg effect, where there's increased levels of glycolysis, right? That does not, even in the presence of oxygen, right? It still is increased in this process. Okay? Additional pathways that lead to using of alternate molecules for producing energy is associated, in these aggressive cells by increased uptake and utilization of TCA cycle intermediates. Another landmark sort of wave, further wave of metabolic reprogramming is the increased uptake and use of amino acids as carbon sources for energy production. And those intermediates can feed into, the TCA cycle. In particular, aggressive cancers use the amino acid glutamine, and they uptake it at higher levels and they metabolize it. And so this is important in the progression of aggressive cancers, and this term for increased use of glutamine is known as a glutamine addiction. Okay? And so that can lead to increased intermediates for the electron transport chain and oxidative phosphorylation. Previous and recent studies have shown that when you inhibit components of oxidative phosphorylation, such as components of the electron transport chain, right, that can suppress aggressive cell proliferation, and it can also sensitize aggressive cells like TNBCs to chemotherapies. So at this point, these 2 points especially in metabolic reprogram seem to be important targets for developing new classes of cancer therapeutics and inhibitors. So targeting metabolism can not only sensitize cells to chemotherapies, but also potentially sensitize cells to NF-kappa beta inhibition. So next, I want to give a brief perspective on some of the important, milestones that plant-derived compounds have played in the development of chemotherapy and anti-cancer agents. So across all different cultures, around the world, there's a rich history of using, locally available plants for medicinal purposes. And here in the United States, one of the earliest, recognized or documented, sources of plant-derived therapeutics was the use of this Podophyllum peltatum root by the Native Americans. So this extract from these roots, right, was used as cathartics, right, antiparasitics, purgatives, and even anti-cancer agents. So over several decades, right, after its first identified uses, there was several efforts, right, formalized efforts to isolate the active components from this root extract and then start to test their anti-cancer properties and essentially develop, molecules based on these natural compounds. So the earliest studies by John King were able to, improve extraction purposes, right, for isolating the resin from this plant, to test its biological activities. Okay. Further studies by Podowostowski was able to crystallize the bioactive ingredient that they called podophyllitoxin, responsible for biological activities. Further studies after that showed that this podophyllitoxin could be an effective anti-cancer agent in vitro and in vivo. Studies by Stalin and Val Wartburg developed single molecule inhibitors, so topoisomerase inhibitors that were synthesized based on, this toxic agent from this plant root. So around this time, of developing this anti-cancer agents, the National Institutes of Health in the U.S. established a National Cancer Institute, which began large-scale screening of natural products on cancer models to isolate its bioactive components. So in these early studies associated with NCI, right, some of the plants that were first studied were this C. rosaceus, and the compounds isolated from this medicinal plant led to the identification of the vinca-alkaloids, vincristine and vinblastine, which are, anti-mitotic agents, against breast cancer or against cancers. Okay. Further studies by Wall, Wani, and at Research Triangle Institute were able to identify camptothecin and taxane, which are all other classes of effective anti-cancer agents that are topoisomerase inhibitors and mitotic, inhibitors. Since this studies in the mid-50s, more recent studies have identified other plants that have anti-cancer activities, particularly noscapine, curcumin, berberine, and parthenolide have been shown to have anti-cancer activities, from their extracts and components. And also, there was a discovery of anti-cancer actions in resveratrol, which is an agent found in grape skins. So interestingly enough, these compounds that have been more recently studied have been shown to have multiple effects on cancer cells. In addition to affecting, mitotic, or proliferative actions, it affects in cell invasion, apoptosis activities, and inflammation activities. So continued studies of these medicinal plants, right, has been shown to be, very valuable, continuing to be very valuable for identifying new classes of natural compounds for cancer drug discovery. So now let's turn to the plant that we've been interested for the last couple of years, Lippia origanoides. So this is known as mountain oregano, which is, grown or present, has been identified in Central and South America. Okay? And there's over 200 species actually of Lippia, and we're particularly interested in this Lippia origanoides and identifying its medicinal properties and potential anti-cancer actions. So extracts from several species of Lippia have shown to have anti-mutagenic, anti-inflammatory, and anti-proliferative effects. So these studies, right, have specifically indicated the actions of these various species of Lippia, and these species of Lippia have been shown to have anti-cancer actions in, primarily colon cancer and liver cancer. So interestingly enough, right, components that have been identified, right, in Lippia origanoides have been shown in different aspects in different studies to identify NF-kappa beta signaling. So this table lists several of the primary or most abundant compounds found in Lippia origanoides. And in each of these studies, you show in one aspect or another in these different, studies and cell lines that they affect NF-kappa beta signaling at different points. So knowing that the Lippia origanoides, right, can affect NF-kappa beta signaling, in different systems, right? And so we were very interested, and we know that NF-kappa beta signaling is associated with aggressive cancers, right? Our hypothesis was, is Lippia origanoides in particular, the extract, will it inhibit NF-kappa beta signaling to induce cell death in triple-negative breast cancer cells? Right? So in this presentation, I want to really hone in on our studies that tested the abilities of Lippia origanoides extract to induce apoptosis in TNBCs in vitro, and I want to share some of the broad scale insights into the mechanisms of action of Lippia origanoides extracts on TNBC cells. So in this first part of our studies, we initially began by treating triple negative breast cancer cells, MDA231, and normal cells, MCF10 cells, with increasing concentrations of Lippia, or a control treatment. So these are representative data from our cancer cells, TNBC cells, and normal cells. We actually did test several different cell lines that were in this category of TNBC, and several different cell lines that were representative of normal cells. And this data, is representative of all those different tests that we did. So in short, when the triple negative breast cancer cells were treated with increasing concentrations of Lippia over 1, 2, or 3 days, we see a significant, a substantial reduction in cell viability, across concentrations of Lippia. Even at the lower levels of Lippia, we saw a huge reduction in viability over time. Compared to treatment of Lippia, of normal cells with Lippia. So across the majority of the concentration used, there was very little effect of Lippia on normal-like mammary cells. Only at the higher levels did we see, a reduction in viability over the different time courses. Now, first thing we wanted to do after that was to try to get a better understanding of why the viability was going down. So we started to measure different markers of apoptosis, right, in these Lippia-treated TNBC cells. So we did a FACS analysis to quantify annexin and quantify cell death, right, in MDA231 treated with Lippia. And essentially, when we quantify the data, we see here that compared to vehicles, right, at the higher levels of Lippia, we saw a dramatically reduced amount of living cells, and we saw a great increase in cells that were either in early or late apoptosis. So it's important to note that there was very little amount of cells that were necrotic in response to even the higher concentrations of Lippia. So this is indicating that Lippia is promoting this programmed intentional cell death, and not a more toxic sort of cell death that's known as necrosis. So that's important when considering that we want a treatment that's going to be selective and that's not going to be harsh on all the cells in the body. Next, we wanted to analyze other specific markers of apoptosis, particularly caspase-8 and PARP cleavage. So we measured this by Western blot analysis. Okay, so we treated the MDA231 cells at short or a little longer time periods with increasing amounts of Lippia. And essentially we showed that at the longer time periods with high levels of Lippia treatment, there was a substantial increase in caspase-8, which is associated with increased apoptosis. And also we saw increased levels of the protein PARP, which is also associated with apoptosis. In addition to these markers, we also tried to get some insight, or we gathered some insight into cell cycle arrest associated proteins, particularly Cyclin D1 and CIAP2. This is also measured by Western blot analysis in MDA231 cells treated with Lippia at the higher concentrations at 12 and 24 hours. And at the higher concentrations over the longer period of time, we saw that Lippia caused a significant reduction in both CIAP2 and Cyclin D1. In the third segment of this initial characterizations of the action of Lippia, we wanted to get some insight into how Lippia may be affecting NF-kappa beta signaling. And so we measured levels of RIP1 by Western blot analysis. So this is the initial component, so upstream of NF-kappa beta, signaling molecules. And so we showed that there was a substantial reduction also of receptor interacting protein, right, an upstream component of NF-kappa beta signaling, in response to Lippia in the MDA231 cells, right? So this supports the idea that Lippia extracts induce cell cycle arrest, inhibit major scaffolding protein RIP1, which is involved in the NF-kappa beta pathway. So this first set of studies can be summarized by saying the Lippia origanoides extract induces significant sustained loss of viability in triple-negative breast cancer cells with reduced potency towards normal mammary epithelial cells. Cell death in TNBC cells treated with the Lippia extract occurred through apoptosis accompanied by caspase 8 activation. Lippia origanoides induced G1 interface cell cycle arrest. So this is an additional set of studies of the data that I did not show you, but this was also a FACS analysis looking at the different parts of the cell cycle and showed that Lippia did induce cell cycle arrest at the initial interface of the cell cycle. And that was associated with the measurements that I showed you that had showed the loss of Cyclin D1 and CIAP2 protein levels. So in addition, we also showed a reduction of RIP1, which points to inhibitory effect on NF-kappa beta signaling. So we can say at this point that when triple-negative breast cancer cells are treated in culture with Lippia, that leads to apoptosis through caspase 8 activation. And this is associated with the perturbation of the NF-kappa beta signaling, particularly a reduction in RIP1, and an initial component of NF-kappa beta signaling, and a reduction in these proteins, which is an outcome of NF-kappa beta induced transcription. So when this complex 1 pathway or NF-kappa beta protein is suppressed, right, then that can lead to G1 phase cell cycle arrest, and it can lead to the dominant factor activation of the complex 2 pathway, where TNF alpha binds to its receptor, activates FAD, and then that can promote activation of apoptosis through caspase 8. So that by suppressing NF-kappa beta, you're not only suppressing cell survival and proliferation, but you're also suppressing its ability to suppress apoptosis. So then this complex 2 predominates and further supports apoptosis. Okay. In the second part of our studies, we wanted to get a broad scale sort of insight into the anti-cancer mechanisms of Lippia origanoides, and to do this we used quantitative proteomics. So this is a label-free quantitative proteomics set of methods. Okay. And essentially, this is how the experiments were performed for using this method. So cells were treated without or with Lippia for 12 hours. The cells were collected, homogenized, and then separated into the soluble fractions or the membrane fractions. Once these were separated, the proteins from each of these fractions were purified, and then they were trypsinized to break up the proteins into fragments. Okay, these isolated purified protein fragments from each of the treatments were then prepared by solid phase extraction and drying, and then they were resuspended and used for LC-MS-MS. Okay. So essentially, this generates spectra profiles of each of these fragments, which can give insights into, once these are integrated, they can give insights into the types of proteins that are present under each condition. So when these spectra are fed into the MaxQuant software, the MaxQuant actually assigns a level of fluorescence to each of the trypsinized proteins. And then when those proteins are, reintegrated so that we can identify their respective proteins, then you have an average amount of fluorescence for each of the peptides, right, is calculated that represents a particular level of fluorescence for each protein that's identified. And at that point, not only the amount of proteins or the identification of proteins that are differentially expressed upon the treatments, but the relative abundance of those proteins under each of these treatments. So this allows you to, and the sensitivity of this technique also allows you to identify a lot of different proteins because there's a wide, amount of sensitivity, under this method. So there's some big advantages here. Once the list of differentially expressed proteins is identified, then and they are used or are fed into the software analysis to identify how those proteins are connected with one another in different pathways. So this is generally the GO analysis was used here. In this part of the slide, I'm just, showing some data that uses the spectra to quantify the R-squared values for each of our sets of replicates for our treatments, the L42 treatments or the vehicle control treatments. And this is showing essentially that the R-squared values within, a set of treatments were identical, right? And so in this set of vehicle treatments, there's also, identical. However, when you compare the R-squared values across treatments, right, they are very much different. So this shows you the very high quality, values of our data. So moving up from there, we have some overall, general information about the proteins that we were able to identify using these methods. So we were able to identify over 3,000 proteins, across the 3 replicates. When these proteins were filtered, right, we used over 2,400 proteins for analysis. So basically the label-free quantitative intensities, the fluorescence intensities of these proteins, right, were used to generate the log2 LFQ values for each of the proteins. So when these values were compared from one treatment or another, those were generated or used to generate the delta log2 LFQ values to show how the proteins changed, the identity of the proteins changed, and the amount of proteins changed from one treatment to another. Okay? So when we had these values quantified, basically the proteins with the fold chain values, right, of these quantities that were either lower than a negative 0.5 or higher than a 0.5 value with a p-value less than 0.005. Those were used for our GO analysis, for our gene ontology analysis. Right? So those were our requirements, our cutoffs. And essentially those were used, the GO values were used in different ways to look at our differentially expressed proteins to identify whether the proteins were upregulated or downregulated. And this is a heat map showing that proteins in blue were downregulated when cells, the MDA231s, were treated with the Lippia compared to a vehicle. And in this perspective, we used the settings to identify the localization of proteins that were downregulated, okay, in response to Lippia. And what we saw, a very strong, sort of predominance of an effect of Lippia was that it impacted or reduced proteins that were associated with proteins in the mitochondria. So either in the mitochondrial matrix, the mitochondrial nucleoid, mitochondrial ribosome. So a predominance, an abundance of these proteins that were downregulated in response to Lippia were localized to the mitochondria. Now when we analyzed proteins, right, and grouped them based on their function, we saw some very, correlative type of results. And that is that cells treated with Lippia had a substantial reduction right? A significant reduction in proteins that were associated with functions connected with mitochondria. So whether it be oxidoreductase activity, NAD binding, electron transport activity, or structural constituents of the ribosome, we also saw, big changes, some of the bigger changes associated with mitochondrial function. So this gave insight that the L42 has a substantial impact on mitochondria. So we next really drew out and focused on exactly where, right? When we look at the pathways important for mitochondrial function, we looked at the specific proteins, okay, that were affected by Lippia and where they fit in to the major processes of the mitochondria. So specifically, we looked at enzymes associated with the TCA cycle and those associated with the electron transport chain. And here we show, right, the multiple enzymes and proteins that are associated with the TCA cycle. We know that once glucose is metabolized, it produces acetyl-CoA, which then feeds into the TCA cycle, which produces intermediates that eventually feed into the electron transport chain to produce large amounts of ATP. We know that aggressive cancer cells increase their abilities to metabolize other, precursors, such as fatty acids and amino acids that can then also feed into the TCA cycle. Well, interestingly enough, we see here that at multiple points across the TCA cycle, Lippia treatment of MDA231 cells cause a reduction in several proteins around this TCA cycle. In particular, we saw a big reduction in each of the proteins that together form this complex enzyme, alpha-ketoglutarate dehydrogenase. So this is really important because this alpha-ketoglutarate dehydrogenase is a rate-limiting step in this TCA cycle. So we know that Lippia, affects mitochondria, and in particular, it affects multiple points, right, of the TCA cycle that affect, right, the metabolism of other molecules that eventually feed into the TCA cycle. So this is important insight into why this Lippia, right, has such a significant effect on MDA231, survival. So taking a closer look at another component of the mitochondria, right, the electron transport chain, right, we see that Lippia also affects different parts of this complex, right? So there's 5 complexes, right, that transfer electrons in the presence of oxygen, and eventually they also cause a proton gradient build up, okay, in the intermembrane space. And when those protons cross back into the matrix of the mitochondria, right, substantial amounts of ATP are made from this complex 5 ATP synthase. So in a response to Lippia treatment in TNBC cells, we saw a dramatic decrease in many of the proteins that are associated with complex 1 of this electron transport chain. So this is important because this is also a rate-limiting step in this chain of reactions. Okay? Another thing that we wanted to point out, or that I want to point out, is that many of these proteins that are affected by Lippia, that are downregulated by Lippia, right, are nuclear encoded and are transported into the mitochondrion, right? So this is raises other important points on how Lippia may be acting, right, to regulate these proteins or regulate, transport of these proteins or affect the production of these proteins or degradation of these proteins. Now, one thing that has been noted in literature is that when complex 1 activity is lost, right, that can lead to mitochondrial membrane depolarization, right, and eventually apoptosis. So in the next set of experiments, we wanted to really confirm, right, this mitochondrial actions of Lippia by measuring mitochondrial membrane potential. And so we did this by treating MDA231s, with Lippia or without Lippia. And then we evaluated mitochondrial membrane depolarization by using a fluorescent dye, TMRE. And we can see here in the control cells without Lippia that there was an abundance of this dye binding to active mitochondria in these cells. Okay, so the membrane activity was intact. However, in cells that were treated with the Lippia with L42, we saw a substantial reduction in the dye binding to active mitochondria in these cells. Okay. So here's a quantification of the fluorescent intensity of this dye, showing a loss of membrane or mitochondria action, which is a reflection of membrane depolarization. And previous studies shown that it's well known that the loss of a mitochondrial action and metabolism is known trigger for caspase-3 activated apoptosis. So in summary, right, from these studies, or this second part of the experiments, we can say that TNBC cells are treated with L42 showed a significant loss of critical enzymes involved in mitochondrial metabolism and membrane polarization. That membrane depolarization observed in these cells is a known trigger for caspase-3 dependent apoptosis. Okay. So this leads us, to show, right, or summarize that Lippia, right, can trigger ultimately caspase-8 and apoptosis and cell cycle arrest through inhibiting mitochondrial action. It can inhibit TCA cycle as well as complex 1 in the electron transport chain. In additional studies that we didn't show here, right, we also confirmed that the Lippia can suppress the glutaminase enzyme in these cells. And glutaminase is important for converting glutamine to glutamate, okay, and this is important for cell survival. When this metabolism is suppressed, okay, then alpha-ketoglutarate, right, it cannot be formed. It cannot activate the TCA cycle, right, and cause energy production or formation of lipids and other amino acids. So there's multiple ways in which this Lippia extract is acting, right, to eventually cause apoptosis or G0, G1 interface cell cycle arrest. So to summarize the whole project, we know that Lippia origanoides extract can induce potent sustained apoptosis in TNBC cells to a greater amount compared to normal mammary cells, and that the mechanisms of this action include inhibiting NF-kappa beta, halting mitochondrial metabolism, and mitochondrial membrane depolarization. We can conclude from these studies that our work provides further support for NF-kappa beta and mitochondrial metabolism targeted drug discovery, right, for TNBC, right, that these studies can lead to sustained apoptosis in highly aggressive triple-negative breast cancer cells and reduced effect on normal cells, right, to sort of suppressing or limiting toxicity in vivo. These studies also support the power of omics, novel omics technology to identify complex changes occurring in cells. So these activities or these technologies are definitely more sensitive, and they can give you a broader scale, right, of what's happening in cells upon treatment. They expedite the identification of bioactive compounds and their mechanism of actions, leading to this gives important implications, right, for early identification of off-target effects and identifying valuable new targets for developing therapeutics. Okay, so also, or finally, this provides an impetus for further investigations, right, of medicinal plants, especially of Lippia, for their anti-cancer properties. To finish my presentation, I want to acknowledge particularly Vashak Raman, a graduate student who graduated a year ago that was involved in the majority, implementing a majority of these studies. I also want to thank Dr. Morris Levy and Meches Levy for collaborating and connecting us with our collaborators in Colombia, who are the source of these, initiating these studies and the source of the Lippia origanoides. And I also finally want to thank our collaborators at Purdue in our proteomics facility for performing the quantitative proteomics methods. So thank you very much for the opportunity to share some of our work, and I appreciate your time and interest.